Dear learners, welcome to the today's session on chromatography. Myself, Dr. M. Abdul Karim, Assistant Professor, Department of Biochemistry, Faculty of Science, Indira Gandhi National Open University. Friends, in our previous sessions, we have discussed about the procedures regarding the extraction of proteins from cells. After extracting proteins, we need to go for separating those proteins and as well as the biomolecules that are present in that extract. Okay, once after completing this extraction separation, we are going to perform the analysis part. But however, in this session, we will be discussing mainly about chromatographic techniques. And in this particular session, you will be discussing about paper chromatography and thin layer chromatography. Now, let us see the expected learning outcomes of this particular session. In this session, you will be familiarizing about basic principles of chromatography, and you are going to study about different types of techniques, that is like paper chromatography and thin layer chromatography. And also we are going to discuss about the chromatographic applications. And finally, at the end of this session, we are going to see their utility in our day-to-day activities. So this is about the session. And looking into the background of the session. So why we are going to discuss this particular session here? As I said earlier, since we have discussed about the extraction of subcellular components along with proteins from a cell, now we are going to discuss about how to separate them. Okay, here whenever you perform separation of proteins, it is important to take care of their basic structure. Along with that, we should not disturb the activity. So in these techniques, we will be taking the detailed minute precautions also which are need to be taken care while separating the biomolecules using paper chromatography. Yeah, so coming to the definition of this one. If you look into the term chromatography, so if you see the word chromo stands for color and graphy stands for to write. That means here the result which we are going to obtain at the end of the technique, at the end of the entire procedure of chromatography, the result will be in a colored format. That's why this technique is named as chromatography. And the first technique that which we are going to discuss in this session is paper chromatography. And paper chromatography is developed by a scientist known as Michael Twet in the year 1906. He is basically a botanist. Okay, and he is interested in separating the plant pigments. And I think you all know that plant pigments like carotenoids, okay, and some chlorophylls, he want to isolate them, he want to separate them. For that purpose, he started doing some experiments by using paper chromatography, and finally he succeeded in separating these plant pigments by using paper chromatography. Let us move further to more about this technique. See, any technique, any procedure, if you want to perform, you need to have certain basic requirements, that is basic components. Coming to the components that are required to perform paper chromatography, there are majorly two components. One is called stationary phase, the other one is called mobile phase. Here, learners, it is important to understand the terminology also. What is mean by stationary phase and what is mean by mobile phase? Since we are talking about separation of molecules, here we are going to separate the molecules by using two different liquid phases, you can say for time being. That is called stationary phase, another one is called mobile phase. And since the name stationary, that means it is going to be stable. It is not going to move here and there. It will remain there. But whereas this stationary phase is of different types. Sometimes it may be a gel, sometimes it may be a solid, okay, sometimes it may be a mixture of solvents. Similarly, going to the mobile phase is many a times it will be a liquid or it will is going to be a gas. Okay, now if you just imagine what is happening in this technique, we are going to separate the molecules in between these two phases. Okay, we will know more details when you move further. Okay, this is about the components of chromatography. Now let us see the principle. The principle working behind this technique is, one is partition, another one is adsorption. These are the two fundamental basic principles that are operating this technique. Okay, so as I said, the separation in this technique is purely dependent on the nature of the sample that we are going to separate. Okay, for example, if this sample is able to mix and move along with stationary phase, the result will be somewhat different. And similarly, if the sample is able to mix and move with the mobile phase, the result will be different. Okay, but however, broadly, the major principles are, one is partition, another one is adsorption. Let us know more about these two techniques. Yeah, so for analyzing the separation, we are going to use a factor that is called distribution coefficient. You can see in the equation here, this distribution coefficient is represented as concentration in phase X by concentration in phase Y. So you can say phase X as stationary phase and phase Y as mobile phase. That means the separation of molecule in between these two phases. That is represented with the equation distribution coefficient. And if you see here in this example, we are showing a piece of chalk, which we are going to take this piece of chalk and we are going to dip this piece of chalk in a dye. Okay, even you can try this experiment at your home also, where we are going to take a piece of chalk and dip that piece of chalk into the dye. Okay, after a few seconds, like 2 to 3 seconds, you can remove the chalk and break the chalk and you can see what will happen. The white chalk which you have taken initially is now having a blue color, a dye color on the top of the chalk. Okay, however, in the middle of the chalk you can see a clear zone without any dye. Here in this entire exercise, what we are trying to convey? We are trying to convey that when this chalk is dipped into the particular dye, what is happening? The dye is going to adsorb on the surface. That's what is called as adsorption. So adsorption is a physical process where there is increase in the concentration at the surface area. This is called adsorption. So by using such simple principles, the technique of chromatography is being carried out. Let us know more about this. Yeah, as I said, there are two phases. One is stationary phase, the other one is mobile phase. Okay, so based on the nature of stationary phase and mobile phase, we are going to have different types of techniques. Okay, for time being, I will just let you know the different types of techniques. For example, if you see in this table, the stationary phase, if it is a liquid, then the mobile phase is also liquid, then the type of technique we are going to use is called paper chromatography. And similarly, thin layer chromatography and your HPLC, that means high performance liquid chromatography. All these three techniques are having two phases. One phase is called liquid phase, the other phase is called mobile phase. Okay, and similarly, when you are going to use solid as a stationary phase, solid material as a stationary phase, and the mobile phase, if it is a liquid, then we are going to call it as adsorption column chromatography. Similarly, the support material, that means the solid support, if it is an ion exchange material. See, if you recollect the basics of biomolecules, biomolecules are having different charges. For example, you are going to separate a molecule which is having a specific charge, then you can separate the molecule based on its charge. That's why we are going to use here the solid support, which is having the ability to exchange the biomolecules. And similarly, the another variant of this chromatography technique is gel permeation chromatography, where again we are going to use solid matrix. However, learners, we are going to know about all these techniques in detail one by one in our today's session and as well as in the coming sessions. Okay. Yeah, since in this session we were restricted to discuss about the partition chromatography, let us know more about this partition chromatography. The best examples under partition chromatography are paper chromatography and thin layer chromatography. The first technique, as I said, we will be dealing with paper chromatography. Okay, here, as I said, the principle of separation is distribution coefficient. Okay, here the sample is going to be distributed in between two liquid phases. The first liquid phase is stationary phase, and here since I am saying paper and also I am saying that there is a liquid phase, means here if you just practically imagine, if you are taking a tissue paper and dipping it in water, what will happen? The water will absorb to the paper. Am I right? Similarly, here also we are going to take the paper as a support material, but this paper, when it is going to absorb the solvents, the fiber present in the paper is going to have some moisture, some water. That water will act as a liquid phase. Okay. Similarly, the sample which you are going to use, for example, plant extract, leaf extract or tissue extract, whatever it may be, that solvent, that extract has also got some water in that, some liquid in that. So like this, what is happening? Two liquids are there. One liquid is in the paper, the another liquid is in your solvent, and now there is going to be a separation of this particular sample in between these two solvents. Okay, now let us see about how to proceed further for separation. Okay, to perform this technique we need certain basic instruments and as well as you can say glassware and stationery. Okay, here the major requirements are like Wattman number 1 filter paper. The paper that we are going to use to perform this technique is Wattman number 1 filter paper. Along with this, we are requiring some glass jars. Glass jar, depending on the size of the paper you are going to use. Similarly, we need some solvent system and capillary tubes to load the sample on the paper. And also we will be requiring a spraying gun which is responsible to spray the dye or stain on the paper. Okay, we will also going to observe these instruments in our coming discussions. But however, since we are talking about the solvent system, which is nothing but the mobile phase, that is very essential for this chromatography technique. Okay, in this table you can observe there are different solvent systems. For example, if you see on the extreme right, you can see a composition called butane 1-ol, acetic acid and water, and the ratio is here 40, 10, 50. That means we are going to take butane 40 ml and 10 ml of acetic acid and 50 ml of water, and total will be 100. That is 40 is to 10 is to 50. Okay, and this particular composition we are going to use to separate the amino acids. So like this, if you look into this table, you can observe different types of solvent systems are there. If you want to separate monosaccharides, disaccharides, if you want to separate plant pigments, there are different solvent systems. Why we are having this many different types of solvent systems here? See, again you need to recollect your biomolecules. Why? Because these biomolecules are of different types like amino acids, proteins, carbohydrates, lipids. Okay, so depending on the nature of sample that you are going to separate, you need to choose the solvent system. Okay, So while doing practical experiments, you will learn more about the significance and how to prepare this solvent system. For time being, you try to remember that there are different solvent systems for different types of biomolecules to be separated. Okay? Yeah. So coming to these basic requirements. Basic requirements, as I said, if you see in this picture also, you can observe that there is a glass jar. Okay, and apart from this glass jar, in the middle you can see there is a hanging paper. That's nothing but your Wattman #1 filter paper. And at the bottom of this glass jar, there is a colored solvent. Just for the sake of convenience, we are showing the different colors here. Okay, it is not should that always you will have a colored solvent. Okay, just for color distinguishing, for observation point of view, we are showing different colors. Okay, and here in this example, what we are trying to convey, the entire system, entire basic requirements of the chromatographic system. Yeah, in this slide, you can see the next slide is about ascending and as well as descending chromatography. If you look into the first image, that is descending chromatography, the major difference between the descending and as well as ascending chromatography is the flow of the solvent. If you carefully observe these two images here, in one image you can see, that is in the descending, you can see the solvent is moving from top to down, whereas in the ascending the solvent is moving from bottom to up. Okay, and in both the systems we are having the same paper, same glass jar. Only difference you can clearly observe that the sample spot we have mentioned there. This sample spot is at the top, that means towards the solvent system in the case of descending chromatography, whereas in the case of ascending chromatography the solvent is at the bottom and the sample spot is also at the bottom. In both the cases, when you are going to allow the solvent will move along with the paper. And here it is important to note that the sample spot is a spot where you are going to apply your extract. That means your plant extract or tissue extract or your mixture of sample. It may be water, it may be. You are going to apply there. When solvent is moving, solvent will dissolve the sample and also move further. Okay, so these are the two basic types of chromatographies in paper chromatography again. That is, one is descending, another one is ascending. Yeah, so in this image here you can see on the extreme left side you can see a spraying gun. So this spraying gun is a small apparatus, you can say, which is just nothing but a water pump kind of thing. Okay, so in this spraying gun we are going to load a different dyes, different stains that we are going to put over the paper. Okay, so what will happen when we are going to spray, the paper is going to develop different colored spots. Okay, on the extreme right of this image you can see a developed chromatogram. That means earlier in the beginning, at the beginning when you are taking the paper is plain without any spots. But once you load the sample and allow it to run, after that you are going to have a developed chromatogram which is having different colored spots. Now you need to recollect the definition. Chromatography, chromo means color, graphy means to write. Here you can see the result is in the colored form. Okay, that's why it is named as chromatography. Okay, however, as a part of procedure, once the paper is having a maximal level of the solvent front, then we are going to remove the paper and dry it in an oven. Then we are going to spray the stain over the paper, that is called the staining region. If for example in case of amino acids we are going to separate, then you are going to spray with ninhydrin. Okay, similarly if you are going to separate any other molecule, you are going to use the different stain for that particular sample to be separated. So this is up to the separation part. Once the chromatogram is ready, we are going to identify the spots. That means here there is a small formula which we need to apply, using which we are going to obtain the specific component, because once the color is developed, you don't know which color representing to which molecule. Am I right? So to overcome that, we are going to have RF value. So RF value is a value which is giving between the distance moved by the compound from the origin and distance moved by the solvent from the origin. Okay, by taking these two values and representing in this equation, you will get a specific factor and specific value. That value will help you to understand the particular molecule that has been separated in this technique. Here in this table, you can see different RF values are there. Okay, for different amino acids. As an example, I am representing only amino acids here. However, each and every amino acid has got its different RF value. Okay, so how to obtain RF value? The distance moved by the solvent divided by the distance moved by the solute. If you make these two, you will get a RF value, and whichever the RF value is matching with the particular type of amino acid, you can say that this spot belongs to this particular amino acid. This is how RF values are going to help us. However, since we are talking about paper chromatography, let us just know about the variants of paper chromatography. However, there is ascending and descending. Okay, and apart from that, here there are one more type that is called circular, or you can also call it as disc paper chromatography, where in this case the paper which we are going to use is in the circular form and the rest of the procedure remains same. Okay, this procedure is also rarely used in small laboratories where there are no what is called as different mixture of samples, and when they want to perform a small experiment, they can use this paper chromatography. Now friends, now let us move to the another type of chromatography technique, that is thin layer chromatography. Okay, again here the name itself indicates that thin layer. In the earlier case, we were using paper as a supporting media. Okay, but here in this case we are going to use a thin layer. Thin layer of what? Thin layer of silica, thin layer of alumina. Okay, there are different materials that can be used as a thin layers. Okay, and this technique developed in the year 1938 by a pair of scientists from Russia. One is Asmelov and Shabad. These two combinedly developed this technique. Okay, and here this technique is also based on a principle called distribution coefficient. Let us know more about this technique. Okay, as I said, this technique, the thin layer, we are going to use different materials. You can see there are different materials starting from silica gel. There are again in silica gel we have different ranges from 60 to 120. This is the size of the silica gel. Okay, and alumina, cellulose, magnesium, silica. Okay, and there are so many materials. Okay, similar to paper chromatography, here we also need certain basic requirements. For example, like glass plate, okay, and a spreader, capillary tubes, micropipette, okay, and a TLC developing chamber. So when you see this technique, here the main skill is required. Some skill is required to prepare the TLC plates, that is thin layer plates. This is nothing but we are going to take a glass plate here. Okay, on this glass plate we are going to pour the slurry. The slurry is made up of a powdered support material, okay, by dissolving in water. For example, we are taking silica powder, dissolving in water and pouring on the glass slide. Then you need to spread it with the help of a spreader to get the even thickness. However, in the market nowadays there are ready-made plates are also available. If you are interested, you can prepare and use it. Okay, and the important point to be remembered here is the thickness of this layer depends on the type of technique that you are going to perform. For example, you are going to perform an analytical technique, then the thickness of the layer will be ranging from 0.1 to 0.25 mm for the purpose of analytical. Whereas if you are going to perform the preparative TLC, then you will be taking only 2 mm thickness of the TLC. Okay, and the main thing is once you have spread the plate with this alumina or silica, then you are going to activate the plate. For this activating purpose, we are going to place these plates in the oven for 1 to 2 hours at the temperature of 100 to 120 degrees centigrade. What will happen? By putting these plates in the oven, what will happen? The slurry which we have prepared is going to fix on the TLC plate. That's what we call it as activation of the plate. Okay, and then we are going to now load the sample. That means here the TLC plate is ready. Now at the any of the edges you can take, and you need to make a small hypothetical line, imagine, and then you are going to apply the sample with the help of a capillary tube, or else you can use a micropipette also. Even you can use these wire loops can be used to apply the sample on these TLC plates. Okay, so now once the plate is ready, plate is activated, sample has been applied. Now you need to ready your what is called is TLC chamber. Similarly, here also we are going to use different solvent systems, and we are going to take the solvent system in a glass chamber, and into the glass chamber you are going to introduce your TLC plates. Once you look at the figure, here you can understand the entire setup, how it looks like. So in this image you can see there is a huge glass chamber. In this glass chamber at the bottom you are going to apply the solvent. The white plates are nothing but your activated TLC plates. Now what is happening? Here in this case always solvent moves from bottom to top. Okay, so here the solvent will allow, along with the solvent flow, it will take the sample also. Sample will also move further. This is how the entire TLC setup looks like. Yeah, this sketch also explains you how to load the sample and where is the sample spot and how to run the TLC system. Okay, to obtain the better results in TLC, it is always advised to use constant temperature. Why? Because the temperature will affect the solvent flow. Okay, hence to obtain the repetitive and constant results, it is required to maintain the constant temperature. Okay, and the thickness of the slurry, or you can say the thin layer, need to be uniform without any gaps and without any cracks. Okay, and the final thing is we need to after once the solvent flows through this TLC plate, you need to dry these plates by putting in a hot air oven for 2 to 3 minutes. So by that what is happening, your sample is going to be ready, your TLC plate is going to be ready. Then what we are going to do, we are going to detect the sample. Similar to paper chromatography, where we are going to use this spray, what is called as spraying reagents and stains. Similarly, here also we are having different detecting techniques. For example, if the sample is having what is called your basic biomolecules like carbohydrates, then we are going to use 50 to 25% sulfuric acid in ethanol, which we are going to spray on that. And if the sample is having fluorescence in nature, for example it is having any colored product, then you can directly put this plate under the UV light, where you are going to observe the fluorescent images. In the picture you can see the fluorescent picture, how they illuminate once you apply the UV light on this TLC plate. Okay, this is one way of detecting the sample. And similarly, there is another method where you can even apply iodine vapors to identify the fatty acids, because we have different biomolecules, carbohydrates, proteins, lipids, nucleic acids. For different biomolecules we have different ways of detection. Okay, and similarly if you are using any radio labeled compounds, then we need to expose this particular TLC plate to auto radiography. However, Here also we can apply the same RF formula, that is RF value formula, to obtain the RF values for different amino acids for their easy and accurate detection. Okay. Okay, the another advantage of this technique is, this TLC technique is, if we want to separate, that is fine enough, very good, we are going to separate. But after separation, if you want to quantify how much sample is there in the particular spot, then what we can do, we are going to take that particular spot, we are going to scrap it and dissolve in a suitable solvent, then go for the estimation by using your spectrophotometry and calorimetry or fluorimetry. By using any of these techniques, we can even quantify the sample that is present in the particular colored spot that is developed during thin layer chromatography. Okay, when you compare, if you ask me what is the advantage of this thin layer over the paper chromatography, I would say this thin layer is faster and better in separation and accurate. And apart from that, there are various choices for the stationary phases based on which you can use different methods and different solvent systems and different stationary phases depending on the nature of the sample you are going to separate. So these are the few advantages of thin layer chromatography over your paper chromatography. Okay, yeah. So coming to the sum up, now we are going to sum up the entire discussion which we had today. Okay, so one thing is, in the beginning itself we came to know about stationary phase, mobile phase, okay, and other different principles of this one. And here we have already discussed about the various requirements, the basic requirements to perform both paper chromatography and as well as the thin layer chromatography. And we have already discussed and covered about the various methods of detecting, that is identifying the sample that we have separated in these techniques. And also we know about the applications of this paper chromatography and thin layer chromatography. However, if you want to know more about these two techniques, you can go through our IGNOU self-learning material that is available at GAN course, and there are several other books and references, okay, that will help you to know more about these techniques. Dear learners, thank you one and all, and best wishes.